Good evening, everybody. It's a pleasure to invite Professor Greg Gibson, who's visiting us, and he has kindly accepted to give a lecture on a subject which is very, very exciting, and it's been a wonderful 2 days interactions we've had with him, and he's a professor of biological sciences and director of the Center of Integrative Genomics at the Georgia Institute of Technology. Raised in Canberra, Australia, he received his BSc in genetics from the University of Sydney, and then
give my talk if you like.
No. And then moved to Biocentrum of the University of Basel in Switzerland for his PhD. After postdoc at Stanford, he moved to the University of Michigan as an assistant professor, then became a member of the quantitative genetics unit at North Carolina State University, during which time he helped to bring the tools of genomics to Drosophila complex trait research. In 2009, he moved to Atlanta and switched to human genetics, developing his interest in genotype by environmental interactions and transcriptome profiling for personalized medicine. He is an elected fellow of the American Association for the Advancement of Science, has authored 2 textbooks and a trade book, It Takes a Genome, and from 2024 will be the new director of the acclaimed Summer Institute of Statistical Genetics. Last year, he was named Regents professor as well as Tom and Mary Patton Chair at Georgia Tech, and he's also an adjunct faculty of BSB, and I've known him for the last 10 years, have had wonderful interactions. He's one of the persons who has championed or believed in me when we started Ayur Genomics. I happened to meet him after a decade of research in Ayur Genomics, and he was very fascinated, and every time I do this research, I keep on catching hold of a person from western part of the world to see how, if I sound it to them, how they will take to it. And it's been an interesting a dinner time interaction in a conference which he visited. That's how I got familiar with him, and he's been mentoring me in different ways in the genomic science area, and he kindly consented to also be here in our department as an adjunct professor, so we will benefit greatly by the new initiatives we are taking, and we are looking forward to his lecture today. So over to you, Greg. Thanks.
Thank you so much. Is that on? Is that better? Yeah. Hi. Is that better? Is it working? Yeah. Okay. I was at a play in London a few days ago, and they actually had their microphones sticking down their heads like that. It was very distracting. So thank you so much, Vitaly, for the wonderful introduction for 2 splendid days. I've really, really enjoyed Jodhpur, what it has to offer, and the hospitality, and the friendship. And I look forward, I hope it's not this first, but I very much hope it's not the last time I come, and I'm looking forward to all sorts of interactions in the future. So this is a talk I've not given before, but it sort of reflects some interests I've had for some time. The fact that we're in the cusp of a real revolution in the way people think about medicine. So the term personalized genomic medicine means different things to different people. In fact, on the way here, as I was at a meeting in Lucknow of the Indian Association for Medical Genetics, where they're doing precision diagnostics of children newborn with congenital conditions, developmental delay, craniofacial abnormalities, and things like that. And I was just astonished to see how rapidly this technology is being adopted here in India to help, as many as 1% of children who are born with such abnormalities. the other side of personalized medicine is the sort of thing that affects all of our lives, and that is trying to predict what sort of diseases we're going to succumb to and see if we can do things either individually or as a population to intervene in that. And what I'll talk about today is a little bit about the hope for that, but mostly about some of the issues that arise given that the fact we're working with an extremely heterogeneous and unique population anywhere in the world, particularly here in India. So my starting point is the observation that actually disease is remarkably geographically distributed. And so this is an obesity map of prevalence across the United States taken in 1990 and then again in 2010, just 20 years later. And you'll notice that in both cases there's what we call the stroke belt across the southeast from Texas through Tennessee and Appalachia, where there's a much, much higher prevalence, in this case, of obesity. Almost, and what's causing that? Is it diet? Is it ancestry? So much more African American populations here. Is it culture? And then, you know, it's also temporal. Notice this, that this is the highest prevalence, was 15 to 20% in 1990, and then it's actually 20 to 24% is the lowest prevalence 20 years later. So it's not just America either. So this is the UK Biobank data. So this is a big study of 500,000 British people who've enrolled. And this is just showing that for rheumatoid arthritis, you see, I think my laser's dying, but that, you know, rheumatoid arthritis is common in Wales and the Midlands, but almost absent in London and Edinburgh, bizarrely, whereas chronic obstructive pulmonary disease is very common where you might expect more industrial country in the Midlands, but relatively rare down here. So disease is structured wherever we look. It's also clearly temporally evolving, and this is the disease I do most of my actual applied translational research on. It's inflammatory bowel disease, or Crohn's disease and ulcerative colitis. And remarkably, the first case is reported in 1859, so just about 150 years ago, and it rapidly increases to around about a stable plateau of 1% throughout the Western world. And then just 50 or 60 years ago, it starts appearing in China and some other countries and increases. And so we don't know either what the delay is or what the cause of that dramatic increase and then hopefully plateau is in this. But we can see that actually it's distributed globally, and India is among the countries that we're seeing an acceleration in incidence. And obviously, if we could identify the genetic and environmental factors causing that, hopefully that would help us in dealing with it. Okay. So then the question is, why do we get sick? What's causing all of this geographically and temporally structured disease? And so I'm going to walk through sort of 4 possible sets of explanations and then focus on the last one. All right, so the first obvious cause is that we in the world are constantly trying to separate ourselves from out there, okay? And the first level of that is that we face a constant barrage of infectious pathogens, whether they're parasites that cause malaria, bacteria which cause things like tuberculosis and pneumonia, or viruses like influenza, or the sort of noroviruses that cause diarrhea, and obviously most recently COVID. That constant infectious disease exposure has been the major source of mortality and morbidity for humans for millennia now. But we're also trying to distinguish ourselves from the abiotic world, from things like pollution, from environmental toxins, from industrial toxins more and more, from the process of aging, and from just the stress of life itself. And so we're constantly inducing an inflammatory response, which is related to the immune response, and it's just constantly chipping away at our well-being. In fact, if I talk to medical colleagues at Emory, many of them sort of believe that inflammation of different organs is what makes us age at different rates. Certainly, there is this argument called the hygiene hypothesis that in this pristine world that we've created in the West, the glass houses we live in, that our immune system is primed to overreact to insults because it doesn't get appropriately adjusted to microbes when we're infants, and there's this big concern that post-COVID, something like that may happen and cause a dramatic increase in autoimmune disease because the immune system has not been appropriately primed. Now, the good news is that there's no hard data that that's going to happen, but the bad news is there's a lot of theory that says it might, and there are hints coming from hospitals in the United States, at any rate, that respiratory diseases are increasing, and so that's something we need to keep an eye on in the coming years, the idea there being that our immune systems have not been appropriately primed to deal with the pathogens that they're supposed to. And then, of course, there's our second genome, what many people call our second genome, which is the microbiome. So humans are 1 part human, 99 parts microbe by number of cells. Those microbes have some genome content that is much, much larger than the human genome. And it's doing important things commensally, helping us to digest foods, but also it's clearly prone to cause damage. It's important for immune modulation. In fact, mice in the lab are in many ways a poor model for humans because they don't have an appropriate microbiome as much as anything else. We know that the microbiome can encourage autism and schizophrenia and affect mental health. And if it can do that, why can't it affect all other sorts of pathogens? Okay, so we're in this constant balance, again, between self and non-self. Now, the good news here is that, we're in an epidemiological transition, and this plot is showing 8 standardized years of life lost per 100,000 in 2016 on a log scale. And this is the change in that value between 2006 and 2016 from the Global Burden of Disease study. So this is worldwide data. So 72% of the loss now is due to non-communicable diseases, whereas just 20% is due to infectious diseases. And so you see down here the big 4 or 5. So you've got HIV, diarrhea, tuberculosis, malaria are all declining in incidence globally, whereas at the top here, the ones that haven't changed, in fact, dengue has gone up, but Parkinson's, cancers, Alzheimer's disease are all stable or increasing. So that's what we call the epidemiological transition of disease. So that infectious component is... So the second possible set of explanations is what I call God's will. So there are strains in America that believe that we are rewarded in life in proportion to the effort. That's the meritocratic principle. Okay, you work hard, you will do well. And you are seeing more and more people saying that we don't need health insurance, for example, because if you only worked harder, you would be protected. That to me is a crazy idea, but it's a strain that you are seeing more and more, that ill health is a punishment for sloth. Perhaps those also of a more religious bent find some comfort in the notion that it's God's will, part of His plan, that there is suffering in order for us to appreciate life. You would have seen this sort of argument for centuries. Now, obviously, as inquisitive minds, we don't find that sufficient. But we can also appreciate that we need to be able to adjust our minds for the existence of suffering in our lives. So that's not causal, but it's responsive. In the Ayurvedic tradition and other Eastern traditions, of course, we emphasize that ill health is what arises from a loss of balance, in your case amongst the doshas. That if you become too stressed, or if you behave in strange ways, that you can, or if you're exposed to poor environmental agents, that you can be knocked out of synchrony of earth, wind, fire, water, and ether, or the doshas, and that's at the root of all illness. And in fact, I think it's very easy for us to forget today that we are less than 150 years removed from a world where biochemistry was essentially unknown, where neurobiology was governed first by Freudian psychology. At about the same time, Kreppel and others were arguing that there was a neurological basis to Alzheimer's disease just 70 or 80 years ago. So I take it for granted that ill health is grounded in biochemistry and that mental health is grounded in neuronal substrates. But that's very, very new concepts that many people on the planet are just not familiar with. So, God's will remains the cause of illness for a huge part of the human population. The good news here is that, I don't know if you've heard of Steven Pinker. He's an American Harvard philosopher and scientist who's written a couple of books, one of which is called Better Angels of Our Nature, actually arguing that no matter what measure of well-being we look at, it's been improving for centuries. And he puts that down to inquiry in science. And here's an example of this. This is just global age-specific mortality and lifespan from 1970 in red to 2000 in blue to 2016, so 20-year spans in green, and you're seeing this rightward shift of age groups, so people are getting older, and if you look at life expectancy from red in the 1970s to blue in 2016, across all countries of the world, this is for males, but females even better data, that it's just increasing. It's also still a function of socio-demographic influence index, so wealth, but irrespective of wealth, you're seeing this increase. With just one or two exceptions, contemporary America is an exception, that we're seeing a decline in life expectancy due to what's called deaths of despair in the last decade, so deaths due to addiction to methamphetamines and opioids. So the third level, if you're on a sort of more liberal bent, then you might emphasize that what's making us sick are social determinants of health. So rather than focusing on the cause of disease, you focus on the inability to overcome incessant inequalities. And primarily, those inequalities are what we might call the environmental lottery. So all of us are born into a community, a life circumstance, which is not about choosing. And of course, that means we have divergent access to healthcare. And healthcare means early screening, it means comprehensive preventive medicine, and it means access to the higher quality of care. I work on inflammatory bowel disease, and we have pretty similar rates of that in African Americans and European ancestry Americans, but disease progression is much, much worse in African Americans. And we don't know whether that's due to underlying genetics and biology, or if it's due simply to the fact that African Americans tend to be late to access health care, and by the time they have the disease, it's too late for the interventions that would prevent that progression. And I would bet that it's a combination of those, but certainly the social determinants are critical, and it's just as important as anything I'll be talking about in the rest of the lecture. To add to them, there is this massive emphasis in the public dialogue in the West now that there are historical inequities, gender being perhaps the greatest one. Women's health care was given little attention as soon as 20 or 30 years ago. And in fact, one of my students has shown that even though every single NIH grant you right now has to address whether or not you will study male and female samples, only 10% of recent gene expression data sets report male and female, and only a fraction of those actually take it into account in their analyses, even though it's very, very clear that men and women are completely different in critical aspects of health. So that's an inequity. Obviously, race and ethnicity is a massive socioeconomic inequity, as is poverty in rural and working class communities, and you see that all over the world. I don't need to remind you of the inequities due to caste and access to healthcare, but if you go to cities like Glasgow in Scotland, it's well known that the east side of the city has a life expectancy that's 10 years lower than the rest of the city, and that's all related to social inequities. And of course, these days we're also focusing on things like LGBTQ+, disabled people with hearing and sight loss, the elderly, and many others. And so the emphasis is there appropriately. And of course, that also brings issues of migrant health. We know that migrants do better in many ways, but their children can suffer from schizophrenia and other conditions, Crohn's disease among them, at a much higher rate than their populations. But then the migrants have to deal with disease that comes from inflammation and coping with the stress there. And I think that observation right there is something that I'm really interested in thinking about with Dr. Mukherjee here, is about the effect of migration from the Thar Desert into the city on health of populations. The slide I wanted to bring up in this case is from a book, a wonderful book called The Spirit Level by Wilkinson and Pickett, a couple of English academics who've accumulated vast troves of data and produced this stunning sort of result. So they've looked at developed countries in Europe, Australia, United States, and others, and said, is there a relationship between indices of mortality, or in this case, a whole range of health and social problems listed here: literacy, infant mortality, homicide levels, imprisonment, teenage births. These are all the things that Steven Pinker says are reducing historically. And yet they asked the question, is there a relationship between an index capturing all of those and income per person? And there's a slight trend here, but actually USA is the outlier because we have very high income, but very poor outcomes in things like infant mortality and homicides, which reduces that measure. Okay, so there may be some weak relationship, but look at this figure, same data plotted. Okay, so the y-axis is identical, but this measure is income equality. So it's the difference in income levels between the top decile and the bottom decile of the income bracket by country, and that regression has an R-squared of about 0.8. So absolutely astonishing data, and they have it for many other conditions, that actually what's contributing to ill health at the population level is simply differential activity, differential income levels. And it's affecting not just the poor, but also the wealthy. The argument there that keeping up with the Joneses is causing stress and reduced life expectancy and mortality also for the wealthy. Okay? So income inequality is a bad thing in general, and he's got similar data by states within the United States, by counties within England. So you see it at that fine structure level. Okay, and then of course the fourth cause is what we might call the genetic lottery. So that title is taken from a wonderful book written by Paige Harden that came out earlier this year, talking about the fact that the genes we get are as much a lottery as the conditions we're born into. So of course we emphasize the social determinants of health, but that doesn't mean that the genetic determinants of health aren't important either. And of course, as a geneticist, that's what I'm going to focus on for the rest of the talk. Okay? So that has different levels. I started out this presentation mentioning that at least 1% of newborn babies suffer from a severe congenital birth defect and will spend their first hours in a neonatal intensive care unit. And genomics is now able to reveal the genetic basis for that in as many as half the cases. There is something called StatSeq, where they can have a pinprick of blood and have that diagnosis within 48 hours. And it leads to interventions, changes in the quality of care, or advice to parents about staying on life support, things like that, that change the outcomes for as many as 80% of those children for what is a minimal cost. So maybe $1,000, which is a lot of money in our context, but those children are going to, that if you share more genes, your incidence is higher, therefore the genes are probably causing or having a causal role in that condition. Okay? So disease susceptibility is just as much a complex trait as height, or the number of years you stay in school, or your body mass index, or your skin pigmentation. If you want to ask how complex they are, it turns out, so we have 20,000 genes in our genome. 4,000 of those genes are involved at some level in how tall you become. So a recent paper in Nature just a few weeks ago saturated the genetic map of height. 12,000 variants, I'll show you in a minute, are important factors that each are adding a fraction of a millimeter, but there's 12,000 of them. That's a lot of variability, and it's why we're all different heights. Well, diabetes risk, cardiovascular disease risk is that complex, okay? And we'll never understand all of it for most complex diseases, but we're getting closer and closer. And we probably don't need to understand all of it, but height is a trait where we do. Okay? So in the past decade, our ability to dissect and understand the genetic architecture of disease has really brought us to this precipice of precision and personalized genomic medicine. And as I say, I'm just astonished how dramatically and rapidly it's being adopted all over the world, including here in India. Okay? So what is this genetics? So I think most of us in the audience are somewhat familiar, but I'll just very quickly sort of say that our genome is made up of 20,000 genes. It doesn't really matter what they are, they encode proteins, but from my perspective, what the genome is about 3 billion of these letters, A, C, G, and T. That's the genetic code. It's actually wound around in a double helix, so each that A is a T and opposite of C is a G, and it goes on. So there's 3 billion pairs of nucleotides. They're arranged on 23 strings or threads. We call them chromosomes. And then you have 2 of these chromosomes each, one from mum, one from dad. So that's our genome. Every single cell in your body has essentially the same genome, but for occasional somatic mutations. In fact, they're less occasional than I thought a few years ago, and maybe they're important for aging, for example. And every 1,000 of those letters or so, there is what we call a polymorphism or a SNP. SNP or SNP stands for single nucleotide polymorphism. If I was a New Zealander, I had to call it a SNP. Okay, so let's call it a SNP or a SNP. Okay, and you see one, maybe this might be one. So here in the midst of this string of letters is, and of course there we go. No, it's pointing on my hand, not on there. That G there, that red G, would be a site that maybe... Now, most of those 3 million that you have are shared with other people, okay? They have frequencies in the population of about 10 or 20%, or 5%. But so they're what we call common polymorphisms. Then there's a whole set which are unique to ourselves. Every one of us has a new mutation that mum and dad didn't have, that's new in our genome. We call those singletons. Most of them have no functional effect, and so they can drift up and down in the genome, but they remain at very low frequencies. If you are unlucky enough to have a mutation that affects a critical gene, the protein encoded by that, you will have one of these congenital abnormalities. And then, in settings where you have a lot of consanguineous marriage and possibly endogamy, such as in India and Pakistan, then you're more likely to see recessive alleles, which is what you've been taught about in school, but actually in outbred populations is not the major source of illness at all. It's new mutations. Okay? So most of us are lucky enough that the new mutations don't have much of an effect, but 1% of kids, they are very consequential. All right? So our task is to find out which of those 3 million are the ones that are making us susceptible to Crohn's disease or cancer or heart disease. So how do we do that? Well, we've now got ways of genotyping, of measuring 1 million of those on a little chip that costs about $50. So it's possible to do millions of people with that, if you've got enough money. And you can measure them, and then you can do this. You can say, well, let's just count up how many of each of those variants there are between a set of cases of people with a condition and a set of controls. So we do this in very large populations for a reason that will become obvious in a minute. So if we actually measure the A in the controls, and we find that there's 20,000 of them in the controls, and so healthy, otherwise healthy people, and whereas the G, the opposite allele, is going to be 80,000 in this case. So we'd expect in the cases, by chance, if it's not having an effect, that there would be how many? There'd be 200 in the cases and 800 of the G if there's a 1% of the population of cases. Okay, so there's 100,000 there. If there's 1,000 cases of 1%, we'd expect there to be 200 and 800. So what we've observed in this particular test is that in fact there are 250 of the A's in the cases versus 750 of the G's. So there appears to be an excess of the A. So what we can do is just actually, if we multiply the 250 by 80,000 and divide that by 750 by 20,000, we get what's called the odds ratio, and that's actually a measure of the effect size of that allele. And in this case, the value is 1.33. So it tells you that having an A is increasing your risk of this disease by about 1.3-fold. Okay, it would be equivalent to a gene called FTO that increases your body weight by a kilogram or so per allele. That's a large effect. Most of the variants that have been found have effect sizes which are 1.05, so they just increase your risk by 5%, or maybe a fraction of a millimeter of height, or a few grams of weight. Now the thing is, we're doing 1 million such tests. There are 1 million independent variants segregating in the genome, and for that we need to divide 0.05, our traditional p-value cutoff, by 1,000,000 to be sure that that variant would not be more, you wouldn't get that just by random given the entire number of associations you look at. So that means we need for p less than 10 to the minus 8. Okay, we don't deal with that in regular life. P-values we deal with in regular life are 0.05. If you see something that's got a chance of being more likely 1 in 20 times, then we say that's unlikely. So now we're saying it's less than 1 in 1,000,000 or more than 1 in 1,000,000 likelihood. So we're really being stringent and careful about that, and the field put that constraint on, and because of that we have a very, very robust science, and the vast, vast majority of the variants we find at that level turn out to be repeatable across studies from place to place to place, okay? So most of these things are just increasing our risk by a fraction of a %, and that's contemporary genetics, okay? Okay, so if we do that for 1,000,000 controls and 500,000 cases, which is sort of sample sizes we're getting to now. So 10 years ago you did 10,000, now we're doing millions because you get people all over the world to work together. If you do that, well, here's an example, okay? So we have this thing called a Manhattan plot. So it's a plot of the place in the genome from the tip of chromosome 1 down to the tip of chromosome 22. And the y-axis is the negative log of the p-value. So it's just inverted. So very, very small p-values have large negative log values. So these peaks here are places in the genome where there's a gene variant that's associated, in this case, with diabetes. And there's several hundred of them, okay? Underneath that, you're seeing what's called a rare variant, or coding-wise association study. They're places where variants have a frequency of less than 1% are associated with disease, and they're all in coding regions, okay? Now the thing about GWAS is it's really only designed to find common variants. This is new technology that lets you find very rare things as well. The thing is that a variant can cause your disease. It can be Mendelian, but if you're the only one who has it, it won't have an impact in the population that's measurable, okay? So now that we've got very large sample sizes, we can do that for rare things, okay? So you put those all together, we can add them up, and this is how generating what's called a polygenic risk score works. So these SNPs are given numbers, and they're preceded by this RS identifier. So what the idea is, we've got 3 probands here, so 3 individuals with a condition, and one of them has an identical twin who does not have the condition. And the idea is that you start them out each with their baseline risk, might be 1%, okay? And then having this AA variant genotype, so I said there's 2, actually in each individual there are 2. Okay, so if you're homozygous for the A, your risk increases by 10% or 5%, whatever it is. If you're heterozygous, it doesn't change. Heterozygous doesn't change again, then actually this variant is increasing the risk as well, and then this one's decreasing it, and you go through it. So the twin actually should have the same genetic risk as their proband one. So this person did not get the disease and this one did. That could just be by chance, or what I'm suggesting here is that sometimes a particular variant maybe interacts with an environmental exposure. Maybe that you were exposed to a virus. Maybe you had an accident, fell off your bike when you were a kid, and that was sufficient to interact with a genotype to dramatically increase your risk in association with this variant as well. Okay? So they can have the same genetic risk, but only one of them gets the disease. Okay? A proband, another proband, has a different genetic risk because they have different genotypes, but the idea is that in both of these cases, if you go through this exercise, they're probably more than average at risk. So they're out there, but they're not the same. Whereas maybe proband 3 has yet another different profile, but maybe their risk ratio is actually not a whole lot larger. So these two is what, 10 and 8, whereas this one's only 6. So they're all different. You don't have to be the top risk to get disease. And even if you have a top risk, you don't necessarily get disease. It turns out for me, ironically enough, my largest polygenic risk is actually for Crohn's disease. And I don't have Crohn's disease, okay? But there's Crohn's disease in my family, so maybe that's because my high risk is reflecting the fact that there is that risk in the family. All right, so what we can do then is we add up these scores, or in that case we're multiplying them together. We can do it on a log scale. So basically what this is just showing is the number of tall alleles. If we go back 10 years for height, when there were only 20 genes known, now we have 12,000, but when we had 20, you could nevertheless add up the number of tall alleles. And if you're in the top decile, you are likely to be about 3 centimeters taller than the population average. And if you're in the bottom decile, you are likely to be 3 centimeters shorter. As we get bigger and bigger sample sizes and more accurate, that regression gets stronger and stronger, and the explanatory power gets better. So now actually it's just as good as knowing mum and dad's height, and in fact it discriminates within families. So 2 siblings have the same risk because mum and dad, just genetically. But now with these polygenic scores, we can say brother is likely to be taller than his other brother or something based on their genetic risk. So it's kind of amazing. But what this sort of plot doesn't show is that there's massive variance. So the error bars on each point are as wide as the figure, okay? And in fact in both directions. So if I give you a polygenic score and say you're in the top percentile, I could actually derive a different score with a Bayesian method, for example, and maybe you'd end up in the middle, okay? So you've got to be a little bit wary about these scores. So they're stratifying, but they're not deterministic. They were actually what's been used in animal and plant breeding for 2 decades now to improve yield of plants or yield milk production of cows. But there, what you care about is the population average derived from a sire or a dam. In human personalized medicine, you care about you, and we're fooling ourselves if we think they're really predictive of mine. Okay, they'll tell me about my family, but they don't really tell us about me. Okay, so I like to think of it in this context. I told some of the students yesterday, this is a beach in North Carolina, which I'll be sitting on about 3 days from now. And if I sit my chair down here, and I look out to sea, I don't know. There's a 50-50 chance that 2 hours from now, these waves will be up here somewhere and I'm going to have to move my chair. Or maybe the waves will be further out to sea and I'll have to walk further out to go surfing. Okay? Because I don't know if the tide's coming in or the tide's going out. Okay? If I look out to sea and I see a slight swelling here and I start seeing a white cap, and I say I'm going to follow that into the shore, I don't know whether that wave is going to be the one that comes further up the shores than the previous ones, or if it's just going to get subsumed in the rest and die out. But that, whether or not that's going to happen is a function of whether the tide is coming in or going out. If I do that for 10 minutes and follow 100 waves, I can actually get a pretty good idea of whether the tide's coming in or going out. So this is a metaphor for how I see genetics and how we should all see genetics, is that the polygenic scores are like the tides. If you're at high tide, it's likely that you're going to be coming further up the beach. If you're at low tide, you're likely to be going receding. If you're a high polygenic score, it's likely you're going to get diabetes. If you're low, it's the other way around. But it's not deterministic. There are too many other things going on to really make a strong prediction for you as an individual. Okay? Nevertheless, Amit Khera a few years ago and Sekta Therison from Harvard came out with this beautiful paper in Nature Genetics where they said for any condition you care to think about, it's now possible to look at the percentile of your polygenic score, much less likely. So people look at that from a relative risk perspective and say, I want to know that. I want to know if my breast cancer risk is 5 times more than the population average, and if I can act on that, that's useful. All right? And what Amit pointed out is that there's many, many more people in these extremes. So there's 1% of the population that are there because of a rare mutation in, say, lipoprotein lipase, which is known to cause hypercholesterolemia. So that's 1 in 10,000 people. So there's 1 in 100 people at that extreme, which has about the same risk. Okay? So that's why people are so encouraged by polygenic scores, because they can stratify our risk. Okay. Well, the issue is that it's really hard to do this across populations. So why? They only capture the genetic component, and it's easy to lose sight of the fact that environment, culture, environment means culture, it means family, it means diet, and many other things matter as well. I will show you some slides illustrating these. These SNP frequencies drift among populations, so the score distributions drift as well. And that, but note that that doesn't mean that the trait has to differ, although oftentimes it does. So don't draw the conclusion that, say, African American score is different from European score to mean that African Americans are different from Europeans. That's a non sequitur. The accuracy of these scores is a function of the variance they explain, and that varies among populations, so does the performance, but guess who loses out? So all of our scores are developed in Europeans. The accuracy drops, so it means that the further you go away from European, the worse the performance. And of course, the worst performance is in African Americans. And so that has to be redressed in health. And of course, India's pan-genome has been shaped by millennia of migrations and endogamy, and is changing now with that mixture between those groups and with external influences. And we have no idea what to do about that. Okay? And just to hint some of these things is Hini Nagpal, my Punjabi graduate student, now postdoc. She did a study of the UK Biobank, some of you have read, where she said, well, what's the effect of different exposures on these, okay? So environment matters, okay? The other thing is genome structure. So this is a picture from the UK Biobank. It's just showing that we can actually do. And then this is a problem of admixture. So what these are chromosome paintings. So you can all go to 23andMe or to Ancestry.com, submit your DNA as a saliva sample, and then they will plot where each part of your genome comes. This is from my colleague Keane Jordan, plotting African, Native American, and European ancestry in 2 individuals from coastal Colombia and mountain Colombia, where there are different mixtures. And the point I wanted to make is that the variants explained per locus mathematically is this product, so PQ are the allele frequencies, beta is the logarithm of that odds ratio, and both of those can change, and they lead to change in these scores. And as we implement this in personalized medicine, we need to take all of that into account, and we just really are still trying to work out how to do it in admixed populations, okay? Nevertheless, you can do really interesting things. So this is a beautiful paper that came out 3 or 4 months ago showing that you can actually independently measure face shape in different populations. And this was a comparison, so it's been done in Europeans. There's now 250 variants in Europeans associated with different aspects of face shape. So they take the whole face as 3D points, and you can divide it up into regions. And it turns out that the nasal region, where is it? On this plot, the nose here, so each one of these rays is a place where there's a genetic association with nasal shape. Well, you can do the same thing in East Asian populations. So the Chinese biobank is going to be 1 million people very, very soon, the Kaduri Beta Bank. So they can do that, and this is just a facial reconstruction of the average European face and the average East Asian face, and these are where they differ the most, in the red and the blue areas, and you can construct that difference, and it turns out you can make a polygenic score based on these face shape variants that pretty much predicts that difference in nose shape. And about a third of the variants are in common, about a third are Asian specific, about a third are European specific, and the reason is that there's been selection for nose shape, and so the European ones are a high frequency in Europeans, and they're almost absent from the Asian population. So selection, we can now see, is shaping face shape. So I think that's extraordinarily interesting and useful, but of course if you do that sort of thing when it comes to disease risk, you've got to be more careful. If you do it in consequence of behavior, you've got to be super careful, okay? But we can do those sort of games now. But we don't just have to rely on genotypes. So a few years back, when I first got interested in human genetics, Youssef Idaftor, this guy here, was my graduate student on Fulbright from Morocco, and I convinced him to do this study where we went out into the desert and tracked desert nomads, so in tents, moving around every 6 weeks or so, and we did a comparison with slum dwellers in Agadir, the city of Agadir, where he was from. And then he actually decided to actually go to a rural village up in the mountains called Boutroch, of Arab villages up in the mountains. Oh, sorry, Berber villages, Amazigh Berber villages up in the mountain. It took him an hour on a donkey. I'd flown down by that point, but that was fun. And so what we did is we collected not just DNA, but also RNA, the blood for sampling the RNA. And we thought we might find dozens of genes which were differentially expressed between these locations. Well, it actually turned out that a third of those 20,000 genes are differentially expressed between people from those 3 locations. So a dramatic effect of lifestyle on gene expression. I plot that here. We went back a few years later and did 200 people. And what you're seeing, the filled circles are men, the open circles are women, and blue is the Amazigh village. Red is the women in the Berber village who actually stay in the village by day, and the men from that village move into the city to work, and their gene expression profile is much, much more like the profile of the Arab residents of the city, okay? So there's a complex genotype or ethnicity by gender, by socioeconomic interaction shaping the effect of gene expression, the expression of those genes in our immune system, implying that there are differences in immune activity. And I didn't do it then, but I would bet that a lot of that difference is due to microbiome differences of our fourth genome. So then what we went ahead and did was actually, so it turns out those SNPs I mentioned that are associated with disease, what 90% of them are doing is influencing the expression of those genes. And so what it looks like is this would be the protective genotype for disease, this would be the risk genotype for disease, and you can go through the genome and find thousands of places where the risk genotype is increasing expression. And it does it irrespective of where you are, but you get this addition of the cultural, socio-cultural thing of the Berber village changing the expression compared to the city. So you've got that interplay. And if you look at that, there's not what we call a genotype by environment interaction. Those lines are parallel. But if you think, suppose that this gene is responsible for some disease like asthma, and so high expression is associated with asthma, so the risk genotype, then actually who's sick, it's not these people on the right, it's the people at the top of the figure, and they're all the people in red with the risk genotype. Okay? So what that means conceptually then is that the people with the high expression and the risk genotype are the ones most at risk. And so if we combine these technologies, we can actually start to understand what it is about our genetics that's making us differentially susceptible to disease in different populations. And so I expect we'll see a lot of these interaction effects at the disease level, at the population level, that are not taken into account in contemporary genetics. Okay? And now this is just my final slide, or final data slide. This is taken from Erin Connolly, one of my current graduate students. And what she's done is pulled from the internet data sets of different types of immune cells, T cells and B cells. And what you can do now is single cell RNA sequencing. So you can see in a single cell, for about $1, you can sequence all 10,000 expressed, well, not 10,000, but 2,000 expressed genes in that cell, and you can count out how much there are of those. And you do that for a few $100s per person, and so she's just trolled the internet, and what she's doing is actually comparing men to women, that gender bias, and she's doing it across the lifespan with age, and what she's doing here is plotting the proportion of each of these major immune cell types, CD8 T cells, NK cells, Treg cells, and showing that they change with age, but the difference between men and women changes with age as well, the magnitude of that difference. And then you can go in and actually see that within each cell, the number of differentially expressed genes also changes. So older individuals, the number of differentially expressed genes, that this is in these different cell types here, is much greater in women. So women's immune system is diverging among the cell types at a much greater rate than the men's. And so what implications there are for men's and women's health, she can study, and she wants to look at the effect of menopause. My point is that we can now use this technology accessible, and it's becoming cheap enough to do this in studies like looking at what happens to traditionally nomadic people when they move into the city and start getting diseases like celiac disease and Crohn's disease and so forth. And if we're clever about it, we do metabolomics and we do microbiome profiling. We can try to stitch together using integrative methods that whole panomic, that integrative genomic view, holistic view of genomic health that goes so far further beyond than just measuring SNPs in a genome and associating them and giving us this coordinated picture. And then we can also sequence the repertoire of immunoglobulins and T cell receptors and see what antigens they're responsible for that might be promoting autoimmune disease. Okay, so I'm just going to finish with this slide. Why do genes make us sick? Well, there are 20,000 things that can go wrong, right? And we're all getting mutations in them. It's really the okay. I have argued for some time that we have an adolescent genome. So what is it about adolescents? They're still growing up, right? They're not mature yet. And they're pretty uncomfortable in the world they're growing up in. They're trying to make sense of the world. That's the way I see the human genome. It's an adolescent genome. we're about 100,000 years old as a species. The average species lives for about 1 million years. 100,000 is a blink of time. We're certainly not at equilibrium. We're rapidly evolving. And guess what? We're evolving our environment in dramatic ways. We're constantly exposed to electronic stimuli. We're constantly on social media, which creates stresses. We've changed our immune exposures. We've changed our diet. Okay, so our genome, wherever it evolved, really didn't evolve for current modern life circumstances, and I think that that's really helping to make us sick. It takes a genome full of over 100,000 common functional variants, all working us together to keep us alive for 80 plus years, and it stands to reason that our risks are different according to the variants of all of those polymorphic sequences. So selection against Crohn's disease and cancer isn't what shapes our disease risk. What shapes our disease risk is just the fact that these variants are there, they're still evolving to make us fitter, but it's a check and balance, just as the dosha are balanced, our genome's in balance. Okay. You're probably expecting a fourth point, but I'm just going to finish there on this one. It is 3 for that. I want to finish by saying that what makes life worthwhile as a scientist is you work with a wonderful group of people, and I do that. This is my current group, and I just would point out that I have Akshaya, who's an undergraduate, I have Neha, who's a master's student, I have Sini, you saw as my postdoc, I have Paramita, who's a graduate student, and I have Varsha, who is about to become a graduate student. So they're part of my team. These are my Shelties, Sagrado, and Salona, and this is Kira's R2. And I don't know what they're seeing out there, but something attracted their attention. Okay, thank you for your attention, and I'd like to close by thanking Mitali. This is my book, It Takes a Genome, that I wrote about 10 years ago, and here's an inscribed
lecture to hear a very complicated topic, and I think you took us through the whole journey in such a beautiful manner, and I have bugged you enough for asking questions over the last 2 days, but in the audience, people have questions. Anybody? Yeah.
Thank you very much for that.
So there's a mic. Can you?
Yeah, so many complex things, but I was wondering because for the last 20-25 years, we are in the sea of this mobile signals, and probably this is more in urban region compared to the rural region. So is there any study to see whether that has effect, or up to what extent?
You mean electronic mobile signals,
yes,
if there's an effect on mental health?
Yes.
Okay. So at one level, there are studies that look at cancer incidence just from holding a phone up to your ear. That data is a little bit equivocal, but I think there's fairly good evidence that there's some effect there, but that's probably not what you're asking. So, you know, in epidemiology, there are a lot of these ideas, like the hygiene hypothesis for immune exposures, lack of immune exposures, failure to prime our immune system. Another one is the cold chain hypothesis that says that the onset of refrigeration actually preserved bugs that are causing Crohn's disease. And for most of these, there's very little evidence. And so I'd actually add to that category neurostimulation. You know, we argue a lot in America whether or not violent videos, which every kid plays, are promoting a rise in incidence of you know, massive gun violence. Okay, I think the impact of those is much less than the fact that just everybody has guns in America. So the data is equivocal, and yet it's really. But I don't know that we've designed the experiments properly to get it epidemiologically. Okay, so I think the answer is no, but it kind of makes too much sense for there not to be an influence of stimuli. I mean, the fact that, you know, three or four generations, kids knew what a cumulus or a cirrus cloud was, but now they would have no idea, but they can tell you exactly what, well, it used to be what Mickey Mouse was doing, but now I don't know what the latest Pokemon or whatever kids are looking at. But that's one level. But then there's also the stimulation level, this fact that there's this constant power. So I wonder about that a whole lot. Physiologically, just like diabetes is overproduction of insulin when we feast ourselves too much on high fat and high sugar foods, it's hard for me not to see that constant stimulation is affecting our reward pathways, and that the body adjusts for that, and that's pushing us. And I could give a different talk about a topic of canalization, where I think it's almost inevitable that these stimuli are contributing, in that case, to mental health.
Thank you.
So yeah.
No, sure.
You
What-
go ahead
what-
and
go
then
ahead,
he
go
can
ahead.
go ahead and then he can ask.
Good evening, greetings from Jodhpur, sir.
Thank you.
Thank you for such elaborate, at the same time very crisp talk. Having a look at your slide on adolescent genomics, just a small hypothesis has popped up in my head. The abiotic world around us is constantly changing, probably at a pace much faster than the evolution of our DNA.
Right.
So I just want to seek your insights. If I hypothesize today that, let me take plastics for example. We are currently focusing on the research of how we are battling, how plastic is affecting our body or probably the entire living beings around. Somewhere down the lane, wouldn't plastic just become a part of our DNA? Wouldn't that be another element like water, air or something and adapted and something like that?
Yeah. So that's great. I mean, plastic's not going to become part of our DNA. It may be a toxin that damages our DNA. But what you're saying is, wouldn't we become adapted to plastics over time? And I think that's true, but that's going to happen over a period of hundreds of generations. So you will have to come back. You have to wait 5,000 years, okay, which would be fun, but probably not practical, okay? But here's the thing that a lot of people worry about, and that is that the selection pressure on things like that has been dramatically reduced, ironically enough, by the high quality of medical care. So a guy named Mike Lynch has written for a number of years now about his concern that increasing mental health issues are likely to come because of what's called mutation. I'm honored to be an adjunct faculty member here, and this is a wonderful gift. Thank you. Thank you all.